Hello everyone, this is Miss D.A. Marvekar, working as Assistant Professor in the Department of Zoology, Rajarshi Chhatrapati Shahu College, Kolhapur. In last lecture, we have discussed one method of a gene transfer, that is calcium chloride method, which was very easy, which can be used for the bacterial cell, but it is having certain limitation, that is it cannot be used for all the types of cells. So here we are going to discuss another transformation technique, that is electroporation. The term indicates that electroporation, that is with the help of applying electric field, we are generating the pores to the cell membrane so that whatever the recombinant vector will be at another side, it will get transformed into the other side of the cell membrane. Now, how it will happen? So, it will require one particular assembly which consists of the cuvette, which is made up of a glass or plastic. Now, it is surrounded with the two electrodes, that is, one is anode, which is having a positive charge, and the cathode electrode, which is having a negative charge. And so, this circuit is completed. When I will apply the power here, the current will flow in this wire, and so that the electric field will be generated in this area, which consists of the cuvette. Now, before performing the electroporation technique or before applying the power, we have to take here the bacterial cell or the host cell suspension, and it is mixed with the recombinant vector, which we are going to transform into the host cell. Once the mixture is taken in the cuvette, then the circuit is completed and the power is supplied. Approximately 240 voltage can be applied here, and then when I will apply the voltage, so the electric field will be generated. As the electric field will get generated, the cell membrane of the cell will get porous, and it will allow the recombinant vector which is present outside the cell membrane, it will, due to the pores which are formed to the cell membrane, the plasmid or the recombinant vector will get entered into the host cell. So obviously, how much amount of a suspension of a bacterial cell you can use? So approximately 50 microliter of a sample can be used, which is mixed with the recombinant vector. Now here, if you are using the circular DNA and linear DNA, then the transformation efficiency by this method will be very workful or useful for the linear DNA instead of the circular one. And if we are using the volume excluder such as polyethylene glycol, and if we are using the 1.25 kilovolt per centimeter of voltage, so the transformation efficiency will get improved. So this is the general concept where electroporation is the method of the transformation is clearly indicated. Here this is the plasma membrane. It is surrounded by the recombinant DNA. I have to transform this recombinant DNA into this host cell, but it will not allow any single molecule to enter into the host cell. But I have to make this host cell as a transform cell. So when I will apply the electric pulse, it will form the pores like the calcium chloride method. And once the pores are formed, then this whatever the recombinant DNA is present at outer environment, it will get entered into the intracellular environment. Once it will get entered, when I will close the or when I will switch off the voltage, then this pores will get sealed, and whatever DNA has got into the cell, it will remain as it is. So after electric pulse, the pores will get resealed and the gene will remain into the cell. So in this method, it has indicated also exogenous DNA will remain outer environment, and the it will get entered due to the pores which are formed by applying electric field or applying the current to generate the electric field. And so when we are going to proceed for this process, then and when we are applying the current, then the cells which will become competent or it will become able to take the DNA from the extracellular environment into it, then that cell which is a competent one, which is called as a electrocompetent cell. So I hope that you have understand this method. This is very nice method, but it is cannot use for each and every type of a cell. There are certain limitations, and so depending upon the host cell, we can use which method, or which can be we can decide which method is suitable for the specific host cell. Thank you so much.